Hello everybody, welcome back to my channel. This is SyntheoGenesis, and in today's episode we're gonna go through the process of bacterial transformation, how it's done, and how to make it more efficient. So we're gonna use BL-21. BL-21 is non-pathogenic E. coli strain, which has been made especially for these transformation protocols. And because of its efficient gene expression and high quality protein development, it is one of the most frequently used E. coli strain in molecular biology. Though it is extremely hard to modify, and even after thousands of transformations, you may not get even one single colony. So it is 4 a.m. right now. Let's begin our experiment. It is gonna be like 3 days long because first of all you're gonna need to incubate cells, grow them. You're gonna need to replace them into a new growth medium without any antibiotics. To maintain this freshness of bacteria, because it is really important to make competent cells and make them take new DNA. So it is not really a long process compared to another experiment, but it is time consuming. So first of all, we're gonna have to make a minimal growth medium without any antibiotics or other additional elements. We need this medium to grow enough bacteria that we will be able to work with. Taking an old E. coli colonies and trying to transform them is not really a good idea. In order to make transcription efficient, you will need a fresh bacterial solution. For every 100 milliliters of water, I use 1.25 grams of albuterol. Please don't murder me about not using gloves. This is a pre-sterilization period, which means glassware have not yet been sterilized. In this case, glassware sterilization happens right after scaling the albuterol. Right after minimal medium cools down, take one single colony with an inoculation loop and put it into an LB solution. In order to make an efficient amount of bacteria, you'll have to incubate cells for 12 hours. Optimal temperature for bacterial incubation is 37 degrees Celsius. Though room temperature works just fine. Bacterial incubation temperature varies based on which bacteria is used in experiment. After those 12 hours, take 100 to 200 microliters of solution and put it into a new growth medium. This way bacteria in stock solution will remain fresh. Now you have to incubate cells for 2-3 hours. Right after incubation, place solution into a 2 ml microcentrifuge tubes. Please keep in mind that tubes have to contain same amount of liquid and they have to be opposite to each other in a microcentrifuge. If you don't, centrifuge might even break and stop working correctly. Centrifuge cells for 2 minutes on a full speed. In my case, this is 10,000 RPM. Now pour over all the supernatant into a waste container. Resuspend cells into a 100 microliter of transformation buffer. It can be a calcium chloride or either magnesium chloride. Put the tubes back into a microcentrifuge and centrifuge them. After it, repeat the centrifugation one more time. After that, pour over the supernatant and repeat the process again. With the help of transformation buffer, we are killing off all the proteins which might inhibit invasion of new DNA into a bacteria. Now pour over the old solution and resuspend the cells into a newer solution of transformation buffer. So this cleaning process is pretty important in its transformation process, so I would recommend not to neglect it. Separate solutions into two tubes. So one of them will be a control, one of them will be a plasmid transformation. Mark control tube with minus, plasmid transformation tube with plus. Now add 8 microliters of plasmid solution into a plus tube. Incubate both of the tubes into a 5 degrees Celsius temperature for 30 minutes. Put pipette tip right into a liquid solution. And before we go any further, let's talk about the plasmid that we're gonna put into a bacteria. So what we put into an E. coli is small circular DNA called plasmid. And what you see onto a screen is RFP plasmid. RFP stands for red fluorescent protein. It basically encodes for protein which makes bacteria vibrantly red and fluorescent under the UV light. So if everything goes right, our bacteria should be able to develop those types of characteristics. So every plasmid is made out of small pieces of DNA. First, antibiotic resistance gene. When you give an E. coli some kind of characteristics, you also give it a power to survive during the presence of certain antibiotics. That makes selection of modified bacteria way easier. Second, original replication. Original replication, ORI or replicon, enables plasmid to replicate itself inside the host cell. There are numerous kinds of ORIs. Some of them are able to make less than 20 copies, like PSC101. So more hardcover and efficient version is PMB1. which is able to make more than 500 copy. And next one is promoter. Promoter might be considered as one of the most important parts of the plasmid. Promoter basically initiates the process of transcription of inserted gene. Next one is self-label marker, which is just like antibiotic resistance gene, but the difference is that we use it when we work not only with bacteria but other microbes and multicellular organisms. And finally, gene of interest or insertion gene. In this case, this is PRFP, red fluorescent protein synthesizer gene. Well, of course, that's not an end of the list. Well, of course, plasmid contains many other parts, but I think it would be better to make a separate video for it. Now we should get back to a transformation process. So, this is the most important part in a cell transformation protocol. It is called the Hedgehog. This is one of the easiest way to make bacteria take new DNA. You basically stress the cells, break their outer membrane, that enables new DNA to invade into a cytoplasm of an E.coli. Normally we would use hot plate and ice bucket, but hot plate is kinda expensive, so instead of that I will use a hot water and ice bucket. Hot water should be 40 degrees Celsius. This is the temperature when E.coli breaks its outer membrane and takes plasmid in. Now take a tubes and put them into a hot plate for exactly 90 seconds. After that, immediately take tubes from the hot plate and put them into an ice bucket for exactly 5 minutes. And congratulations, you successfully modified life. And now put small amount of albibros into a tubes and incubate them for 2 hours. After those 2 hours, plate the cells onto a petri dishes. One of them will be a modified E. coli and one of them will be a controlled E. coli. In 2-3 days, colonies will appear, and you are done.